Welcome back to the Health Longevity Secrets Show with Dr. Robert Lufkin. Today, we learn how diet, genetics, and epigenetics interact with each other to impact our health and longevity, and how to use this information to design personalized lifestyle interventions. Dr. Lucia Aronica received her PhD from the University of Vienna and has research experience from the University of Oxford, University of Southern California, and University Federico II of Naples. She is a lecturer at the Stanford Prevention and Research Center. She is Genomics Lead at Metagenics Inc. and is the editor of the peer-reviewed journal Life by MDPI. Before I begin, I would also like to mention that this show is separate from my teaching and research roles at the medical school, which I'm currently affiliated. It is, however, part of my continuing effort to bring quality, evidence-based information about health and longevity to the general public. Now, enjoy this interview with Lucia Aronica. Dr. Lucia Aronica, welcome to Health Longevity Secrets.
Thank you for having me, Rob. It's my pleasure.
It's so great to have you on the show. And before we dive into the fascinating work that you're doing, maybe you could tell us first a little bit about how you came to be so interested in this field.
Yes, so I've been in the field of epigenetics for more than 15 years. At the beginning, I became interested in genomics in general. And that's because in the year 2000, when I started my master's studies, the human genome was sequenced. And I thought that was an opportunity for the whole field of medicine to uncover new secrets about our biology and how we can use these secrets to help people live a healthier, longer life. And so I started, I switch career very early in life because I studied ancient Greek, Latin literature, poetry during my high school. There is a high school called Liceo Classico in Italy, where I'm from. And I was in love just with humanities and humanistic studies. I never thought I would venture into science. And but that event, I am also curious animal. So I thought, perhaps I will give it a try. Let's venture into something new. And I will never regret that. It's been a lovely journey. So I first became interested in the field of genomics. And then after completing my master's studies in Italy, Naples, I moved to Vienna, where I again ventured in a new field of genomics, epigenetics, and I started to work on one of the many epigenetic modifications. At that time, it was small RNAs, and probably your audience is aware of what epigenetic is, but
maybe you could take a moment, if you don't mind,
yeah,
and differentiate genomics versus epigenetics, and also the three main modifications in epigenetics would be great.
Yes, happy to do that, because I was so lucky to be able to work on each of those three modifications. So let's break it down and make the complex easy to understand. Genetics describes, it refers particularly to the sequence of the DNA inside our cells that provides instructions to make the protein they make up our cells, and also produce enzymes that catalyze important biochemical reactions in our body. And then the epi, the Greek prefix epi, means on the top. So on the top of our genes, we can find molecular tags called epi genetic marks that modulate the activity of those genes, meaning whether they're turned on or off. And this has remarkable consequences in the way we look and perform at the cell level and also at the system level, so the entire organism. For example, we have the same DNA, the same genetics in every single cell of our body, and yet we can see that the cell of my hair look different from the cell of my eyes. And the reasons for that is that my hair cells have a different epigenetics. So they are using some genes, turning on some genes and turning off some other genes, whereas different genes are turned on or off in my eyes. And so this is one of the many examples of how epigenetics can modify the expression of genotype, the genetics, into phenotypes we can perform, and so that's of course a fascinating field, and it has also many implications in health and disease. And we now understand now that most of the diseases we know have actually an epigenetic component rather than a genetic one. And even things like improving health after a lifestyle intervention. We are not going to change our genetics after initiating a new diet or a new training program. What happens is that many genes are turned on or off, and those genes are responsible of the good effects of building muscles or losing weight that we see when we go on a lifestyle intervention. So the epigenetics is basically another phase of genetics, the flexible side of genetics that allows the environment or our lifestyle to be turned into biology and affect, yes, the way we look, perform, and whether we are sick or healthy. So I was very excited to be so lucky to start my PhD in 2006 when the Nobel Prize for RNA interference was awarded. So RNA interference is one of the many mechanisms of epigenetics. So epigenetic modifications include small RNAs that mediate this phenomenon of RNA interference, and then there are histone modifications and DNA methylation. So in order to understand these three modifications, let's first understand how epigenetic works at the molecular level. So genes can be turned on or off by opening or closing the chromatin. The chromatin is the DNA plus histones. Histones are proteins that are positively charged and help the DNA become very compact in our cells. The DNA is negatively charged, the histones are positively charged, they complex together into chromatin, they mix the chromosomes, and so this makes the DNA very compact. The DNA couldn't fit in our cells. It's more than 2 meters long, 6 feet, and it's fitting the nucleus, which is very small, a few microns. It's like fitting a 20 kilometer long string into a ping pong ball. So histones and DNA are compacted in the cell nucleus, but in this way we can't read the genes. We can't use the genes. The genes are off. So in order to turn a genes on, you need to open that. And that's the job of epigenetic modifications. So histone modifications are modifications on the histones, right? So you can modify the histones with different epigenetic modifications. Methylation is very common, is very popular, but there are so many other modifications. There is acetylation, phosphorylation of histones. And when you modify the histones, you change the electric charge of the histones, and so you can affect the way that the DNA is complex. This is one way. On the other side, these histone modifications also act as a flag that recruits other proteins in the cells, machineries that can open and close the chromatin. So they work with many functions. On one hand, they modify the electric charge around the DNA. On the other hand, they recruit protein. So these are histone modifications of the histones. And then there is DNA methylation, which is the methylation. So it's only the modification is a methyl group, CH3, that is added to the DNA itself, and specifically to cytosines, one of the letters of the DNA, that are located within CG dinucleotides in our DNA. And these are called CPG sites. P stands for the phosphoryl group between the two nucleotides. So we are going through some jargon, but it will be useful to understand and what we are going to address later.
Oh,
the small RNA, just a small RNA that are more complicated. I will not go into detail. I started my PhD like that and are fascinating. But why small RNAs are fascinating? Because they are a sort of mobile epigenetic modifications. They help target either histone modifications or DNA methylation where they need to be in the right place at the right time. And why they're mobile? Because small RNAs are synthesized in the nucleus and then shuttled in the cytoplasm. And during my PhD, I was characterizing how this small RNA works in tetrahymena thermophila, which is a protist that became famous actually because the telomerase and telomeres, the working of telomeres was first characterized in tetrahymena thermophila. It's a beautiful model system to study epigenetics.
So this is fascinating. So it's almost as if is it correct to say the genetics is information that we got from our parents and our ancestors, whereas the epigenetics is experience that is a result of our lifetime experiences and our lifestyle choices and all things that happen to us in our life. Is that a correct way of looking at it?
It's correct and beautiful. For me, it's we know and we intuitively know that we are what we do. We are what we eat, and we are what we do. But now with the field of epigenetics shows that actually this information becomes biology, and the genes can learn from this information, remember and store good memories and bad memories of what we do.
Yeah, and it seems like you have a unique experience in that you've worked both with the small RNA at one point in your career, and then you worked with histone modification, I think, after that.
During my PhD, yes.
Yeah,
yes. And at that time, I was studying, then I moved to Oxford, England, and I was studying the role of histone modifications in DNA damage and cancer. There is actually a link of how our DNA between genetics and epigenetics in many levels. And one of these is that when our DNA gets damaged, which is associated with cancer, this affects our epigenetics. And on the other hand, if we have some problems with epigenetics, this makes us more prone to have DNA damage. And David Sinclair is also is doing some work on this link, which I think is very exciting. And I identified at that time one gene in working with yeast cells, that actually this gene, we also have these genes, and is mediating this connection in yeast cells, and this gene is mutated in many cancers in human. And then finally, now I'm studying DNA methylation, the third modification at Stanford, finally being able to marry my interest for molecular biology and epigenetics with my interest for lifestyle medicine. I myself health nut. During my PhD, I also did an experiment in my own kitchen. So being an Italian, I was born and raised eating a typical Italian diet made of the 3 Ps: pasta, pizza, and pane, which means bread. And so I was, and I was actually fearing fats, dietary fats, because I think this is a little common, especially among women who want to be lean. So I was overall healthy, but as I mentioned, I'm also very curious. So when I started my PhD, I tried change diet, because I read a book by Gary Taubes, Good Calories and Bad Calories. And this book was an in-depth review of the science of fats and carbohydrates. And the book was arguing that actually eating a higher fat diet with healthy fat and lowering your carbohydrates may help you improve your blood lipids and metabolism. Anyway, I tried that. I was just amazed when I took my first blood test after 2-3 months of being on a diet. So my triglycerides went down 3-fold and my HDL went up 3-fold. And for me, it was, I was just curious to understand why. And this was in 2007, when Professor Christopher Gardner at Stanford University had published a study in 350 women showing the same thing. These women were randomized to different diets with different content of carbohydrates and fats. This study was the A to Z study, because they tested four diets, from the Atkins to the Orange, Learn, and Zone. Anyway, when I was doing this experiment in my own kitchen, I realized that there was a study in Stanford showing the same benefits of a high-fat, low-carb diet in women. And I had this dream to look at what happens at the epigenetics when you go on a low carbohydrate diet. And so when I was finishing my postdoc in Oxford, I remember the night, it was late, and I must confess, this is my weakest link. I am up late at night, but sometimes that's useful, because that night I decided to write an email to Professor Christopher Gardner and just spontaneously tell him, look, I know that you are planning a follow-up study to the A to Z study called the Diet Fit Studies. And so he was planning to do a bigger study in men and women with 600 people. And I wrote him that I would love to join the group and look at the epigenetics of people, and specifically the DNA methylation. Why I propose DNA methylation? Because we have an accurate test to measure DNA methylation at the single nucleotide level. So DNA methylation is the best modification to study if you want to measure a difference between, for example, pre and post diet. whereas histomodifications are not very quantitative. So I made this proposal, and when I woke up the next day, Christopher told me, yes, join our group. Can I contact your referees? And I was like, wow. I didn't expect that. And so, and for me it was a big life-changing event, because I had been 8 years in Austria at that time. I was loving living there. I had learned the language. I can speak German. I was attached to the, but sometimes in life you have to shake it up. So this is what actually a friend of mine told me, and then so now I'm looking at DNA methylation before and after a lifestyle intervention, specifically a low carb or a low fat diet that the participants of the new study, the Diet Fit study by Professor Christopher Gardner, are following for 1 year. And the trial has been already completed, but there are many groups at Stanford looking at different data, microbiomics, metabolomics, and I am so lucky to lead the epigenetic analysis and also some genetic analysis coming next.
Oh, that's so exciting. The low fat and low carb diet, how low is it on either side, or what are they going for with those?
Yeah, the study design of the Diet Fit study is unique. There was a limbo phase, initial limbo phase called the limbo, because people were encouraged to go as low as they could with carbohydrates or fat. The target was 20 grams of carbohydrates or for the low carb group, and 20 grams of fats for the low fat group. And of course, it's a very hard target to reach, right? So most people deviated from that, but this was by design, because then after 3 months, people were encouraged to titrate back carbs or fats up to a level that was considered sustainable for life. And the Diet Fit study got many critiques for this reason, because many people in the low carb or low fat space felt, why did you encourage people to titrate back in this way? You missed potential benefits of either diets. And I agree with that. I just comment and reply. I know why Christopher designed the study like that, and it's because there is always a trade-off to making a study that is more impactful for public health, something that is more likely, for the diet is more likely most people will actually do in real life. And this was the Diet Fit study, or a study that is more targeted to answer a scientific question. Is low carb or low fat better for these outcomes? And of course, being a molecular biologist, I would have preferred to force people to eat just 20 grams of carbohydrates or fat for 1 year. That will make me the happiest, because I can measure better and stronger signals. So no questions. I get it. But I also understand, the other perspective. And so I really appreciate the opportunity to have learned all these difficult and facets of clinical research. It's not that simple. So I come from a background of being a molecular biologist and being able to isolate factors. So at the beginning, I thought, okay, this is going to be complicated, but that's the beauty also of clinical research.
Yeah, at least you're able to do it in humans and you kind of get what you get. What methylation signals are you going to be looking at with the DNA methylation? Are you going to be using standard clocks or ones that you look at yourself? And maybe back up a little bit and just if you could introduce our audience or refresh our audience on DNA methylation clocks.
Okay, yes, sure. I will first describe the different projects we are doing, and I'm happy actually to have the opportunity to talk about that, because in this way, perhaps I can, some listeners that want to collaborate on these data sets can reach out to me, because I already did many analyses. So we already have a ton of epigenetic data. There are different projects I'm doing. The first that we already have data, it's about just comparing the DNA methylation before and after the diet on the two different diets. And we see remarkable difference. So the genes that are turned on on the low carb diet or low fat diet are completely different from each other. There is little overlap. There are some usual suspects. For example, we see that on the low carb diet, enzymes that are required for fat breakdown are turned on because there is more fat, and but also different pathways that are turned on or off in the different diets. For example, low carb, there is actually the upregulation of some immune pathways, especially related to natural killer cells. The pathways. And then on the low-fat diet, there are some cancer protective genes that were turned on, especially regarding to colon cancer. Anyway, these are just examples. So there is the DNA methylation analysis, just comparing the two diets and understanding which genes are turned on or off. And this is an important part of clinical epigenetics, because, for example, you may prescribe a dietary intervention that is upregulated some genes more than another, depending on your outcome. One example is also, we know, for example, the ketogenic diet is used sometimes to treat drug-resistant epilepsy. And from an epigenetic point of view, we see that on a ketogenic diet, there is an upregulation of inhibitory pathways in the brain and downregulation of the excitatory pathways. So this is an example to say, okay, we know that this is happening behind the scenes of gene expression, and this makes sense. So this is what the first project I'm working on. And then the second project, that we initiated that then was killed by COVID, and I hope that we can bring this back. So I'm happy to talk now virtually and to Christopher Gardner and then Professor Steve Horvath. So we connected together before COVID because Steve Horvath is the professor who developed the epigenetic clocks that I will explain now, and he developed in 2019 a new clock that was predicting really biological age and mortality, and I connected with Steve and Christopher to test this clock that is called the Grim Age, because it's predicting mortality clock in the diet fit study. We had all the plan, so made these plans, and we also involved the University of Vienna, Professor Karl-Heinz Wagner. I also say hi to him. But anyway, we had this plan, and then this is on the backlog. But I hope that we will also assume that plan. So what are epigenetic clocks? So because we have learned that our environment, our lifestyle can change our epigenetics, and we can measure DNA methylation so on many sites. it turns out that when we measure the DNA methylation of 1,000 sites in our genome, we get a readout of the true age of our cells and tissues, which is called biological age and can deviate from our chronological age. So for example, we can be 40 years old, but then have a biological age that is 30 or 50. Biological age is measured by this, the level of DNA methylation at different sites in our genome. And there are many epigenetic clocks that differ from each other depending on which sites they measure, how many sites, and whether the algorithm that were used to train these clocks through machine learning were trained against to predict by chronological age, so or biological age, which is, for example, outcomes of phenotypes of aging, mortality, cancer, for example, or some blood lipids and markers that we know are associated with metabolic disease. So usually the clocks that were trained to predict biological, chronological age are called first generation clocks. And the one that are trained to predict more phenotypes of aging and biological age are called second generation clocks. And the first generation clock that is more, shows the highest correlation with chronological age is the Horvath clock 2013. And then there are a couple of second generation clocks like the Pheno Age by Morgan Levine and the Green Age by Professor Steve Horvath, the one that we hope we are going to use. So these second generation clocks might be better to look at before and after. So the changing in biological age before and after intervention, because this clock correlate with the marker that change, like it can be blood markers or phenotypic aging, whereas the chronological age clock, like of the first generation, like the Horvath clock 2013, might be better to for the prediction of forensic age. For example, for the forensic sample, for example, if you find a DNA sample and you need to know how old was the person at that time, and this actually there was a legal case where I believe that determining the age of the person was important to determine the sentence, because if that person was younger than 18 years old, the penalty would have been different, right? So, you know, then for forensic purpose, you want to have a clock that predicts better chronologically. It doesn't change depending on whether the that person had eggs or muffin for breakfast. All right.
Yeah.
So,
how did the DNA methylation clocks compare with, you mentioned some other ones, but the glycination clocks or the inflammation clocks? We're seeing more and more type of biological clocks coming. Is DNA methylation, it's obviously your area of interest. Is that going to be the most important one, you think, or are they all looking at different things?
These are very interesting questions. So I also teach a class at Stanford called Longing for Longevity, from Biology to Biohacking. And I teach these topics together with Maddalena Adorno. She's the CEO of a company that is developing xenoblockers for senescent cells, and that can be also another marker of aging. So for your questions, as you pointed out, biological age is everything and nothing. So a biological age could be just a biological readout of living, right? So it encompasses many things. Epigenetics is one of the clocks that we use to measure biological age. There are molecular clocks like the one you are mentioning. It's an epigenetic age or a glycan age, and there are telomere-based clocks, and then there is also an immune age based on gene expression of our immune cells, and many more will come. But then there are functional clocks based on simple blood markers. You can get your blood work done, and because many of these blood markers correlate with biological age, you can have a functional clock that many clinicians are starting to use in their clinic just to estimate the biological age. And then there are even digital clocks, like people based on face recognition. Of course, there is an age of the skin that is visible. So this just to make the big picture. Biological age is a new concept that is used to define how the environment can affect the aging of our tissues and cells, but it's a composite metric, because my skin may be older than my liver if I like to sunbathe every day, but I have a healthy diet, right? So to your point, there is not a best clock. Glycan age, for example, I think it will be very relevant as a signal of probably more glucose metabolism problems, right? There are different signals of aging. And for example, I think one interesting and future directions that biological clock should address is that and this is only anecdotal, but there are many athletes that complain that when they take an epigenetic clock measurement, they look older than they are. And yet they are, supposed to do the right things. And I would love to test this hypothesis. I don't know why, but it can be that training, we know, exercise does cause many epigenetic changes, and some of them are actually they turn on inflammatory genes in the short term and cytokines, and these inflammatory genes and cytokines are important to mediate the adaptive response to exercise that is actually good for us, that at the end, by activating these stress pathways, our cells become stronger. And so it could also be that the epigenetic clocks are capturing that inflammation signal. Right? I'm just saying, that we will need to understand which signals are captured by which clock, and so that we really can more than say we are estimating biological age, we are estimating these component of biological age is more relevant for you.
Yeah. So following up on that, now that we see biological clocks and DNA methylation clocks, investigators are starting to see how lifestyle can affect the clocks like you're doing. And also, I think Kara Fitzgerald just published a nice study on that, where they showed a reversal of biological age, at least by one clock. I think it was Steve Horvath's clock. Then the question becomes, what evidence do we have that reversing biological age is manifested through a biological clock affects outcome and survival? I mean, is it just like getting a Botox injection and it'll make you look younger, but it doesn't really change your outcome necessarily. I guess we have David Sinclair's work with the mice and the Yamanaka factors with OSK winding back supposedly the epigenetics on those to decrease age-related vision in his science paper this year. So is that valid, do you think, or do you know any other evidence of that?
Yeah, so this is an important question, and no, we don't have answers. Actually, even Steve Horvath is cautious about, you know, actually what is the clinical utility now of epigenetic clocks. I think that to establish that utility, we need more studies like the ones by Cara Fitzgerald. So you mentioned she's a colleague, and she demonstrated that after a lifestyle intervention, there was a reversal of biological age and some relevant clinical improvement in clinical outcomes, right? So then we can start to demonstrate that there is an association between actually improving clinical outcomes and biological age. And the more we build this evidence, the more we can also use biological age as a surrogate marker for aging, which is very useful because we can't run interventions for years to wait that people are getting older. And for the second question that you were asking about the experiments done by David Sinclair with the epigenetic reprogramming, and just to give a refresher to your audience, we can actually reprogram cells to dedifferentiate from, for example, hair cells to embryonic stem cells. Embryonic stem cells, so is iPSCs actually, is induced pluripotent stem cells. iPSCs can become any type of cells because they don't have the epigenetic marks to instruct the genes on which genes should be turned on or off. So they have a ton of potential. So this is an epigenetic process, and we can add a cocktail of transcription factor that revert a cell into iPSCs, and then this cell can become any type of cell. And there is evidence that when we do that, there is a rejuvenation, the biological clock goes back. For most clocks, actually not all the one, but anyway, this is complicated, but most clock goes back. And David Sinclair even showed that in vivo, in mice, when you transiently treat the optic nerve cells that of mice that had optic nerve damage with this transcription factor, they're called Yamanaka factor, that reprogram the epigenome, then you have actually an epigenetic rejuvenation. So this is the first step to show this, but we don't know whether this will play out in humans, right? For which outcomes, because these were either age mice or mice with an optic nerve damage. So to get back to your question, I think we still, there is a ton to do to make biological age, clinically useful. But in the meantime, I think including these clocks in lifestyle interventions can first of all help this research further. So I encourage all the clinicians to do that, and then can help us understand which mechanisms of aging are more relevant for which the desired outcome of for a lifestyle intervention. So, and it's exciting also for the participants. I am collaborating with some doctors, for example, that are testing biological age while they are prescribing intermittent fasting intervention. And I am editing a special topic for Frontier Genetics with Morgan Levine and Vittorio Sebastiano at Stanford University, aging clocks in longevity medicine. I'm mentioning this because I'm sure there are many clinicians and scientists in your audience, and we would love to get your submission. I think that the topic is running for other 6 months, so don't wait too long.
Oh, great. Would you like us to put your email in the show notes for them to contact you?
Yes.
Is that okay?
Yes,
and we'll have your
yes,
website
sure.
also on there.
Yes, or yeah.
And yeah, if you're looking for collaborators, in addition to scientific collaborators, are you looking for patient or lay people, participant collaborators also, or mainly right now just scientific collaborators?
For now, just scientific collaborators, just because we probably once we have a new study, then yes, I will let you know, and if you can help me perhaps recruit people, yes. But for now, we have a ton of data, and we have the luxury of sitting on a gold mine and having too many things to do. So we founded a collaborator at Imperial College London recently to look at some of the epigenetic data and with the metabolomics. But we have done whole genome bisulfate sequencing on the biggest losers from the study, and then we are also looking at some epigenetic biomarkers of diabetes and whether they change after the intervention. We have the data. It's just like prioritizing which project goes first is difficult, because in the meantime, there are also other studies that we are working on, for example, testing a ketogenic diet and comparing that with a low carb Mediterranean diet. So of course there are too many projects to work on, and I also need to teach. Well, I only have one life. If you also have some collaborator that can clone me, that could be also another very useful thing to do.
But that's so exciting. So many fascinating projects. In the last couple of minutes, maybe you could tell us, with your knowledge of epigenetics and genomics and high fat, high carb, low fat, low carb diets, what lifestyle choices have you made in your own life as far as diet or exercise or supplements and those sorts of things?
Yes, I am happy to share that. And I think I already shared the first part of the story. So I think the biggest change was trying the low carb diet. And since then, I never went back. So it's more now, more than 10 years, 13 years. For a low carb diet, there are many ways of doing a low carb diet, right? But I just mean really eating whole foods, and this is already low carb, because if you are eliminating refined carbohydrates, pasta, bread, pizza, and refined carbohydrates, you are doing a low carb diet. So, and I think this is a common ground, and everyone can agree on that, whether you are vegan, plant-based, and this is already low carb. And I think this is the biggest improvement that people can see in their diet. I'm also within low carb, I'm choosing more low carb vegetables, so that are those that usually grow above ground. And the reason for that is that they tend to be more nutrient dense compared to the calories and more rich in fibers than, for example, tubers. So, and then protein from a variety of sources, the vegetables, of course, and fish, eggs. I prefer fish to meat. I have little meat just because I was born and raised in South Italy. So it's my taste. It's a preference. And then and I think that this is also important. There is not one way. For example, I am actually in ketosis, but I never track myself. I measured my ketones a couple of times while I was teaching a class at Stanford on a ketogenic diet, and I was in ketosis, but I was not even trying to do that. And it's not a standard keto diet like the one they prescribe you, eat butter, meat, nothing of that, right? It's just eating vegetables, low carb vegetables, fish, olive oil, avocado. And this is important because there are many ways of doing a diet, and there are wrong ways of doing any diet. A keto diet, a vegan diet, there are wrong ways of doing all those diets. So I don't like polarization. This is good or this is bad. It depends. There are many ways to do that. So this is my first probably lifestyle habit, and I feel freedom. I don't count calories. I don't track anything. It's for me effortless and enjoyable to have this diet. So another message I think is important is that what is sustainable is also individual. My diet probably will not appeal for most people, but I truly enjoy it and I will never change it. So telling this is unsustainable I think is wrong, because we are eliminating options for people. We are deciding upfront what is sustainable for people. We want to give people options. We don't want to eliminate options for people. So, and then the second is exercise. So I'm big on exercise, but even in that space, I'm a minimalist. I do the most difficult exercise, but a few of I spend little time on those exercises, and I build my exercise in my routine. For example, I do pull-ups while I'm preparing my breakfast, and that's one day. And then the other day I do push-ups and the combination while I prepare dinner. And then on the couple of days, I go to the Stanford gym. I love that gym, and I do things that I can't do at home, like deadlifts and squats, and I love that. But it's really like 2 or 3 exercises. And I define myself as a lazy person because I don't get much other activity, right? Rather than that, I don't run, I don't go. I would love to do that, but I only have so little time. So it's so possible. It's possible. You know, you don't need. I love taking care of my body, and I enjoy the food I eat, and I think it's possible for everybody.
Yeah. Any supplements that you'd like to share?
I do take vitamin D when I can't be outside in the sun, and sometimes some magnesium. I'm also minimalist with the supplements. I take omega-3s, magnesium, potassium because I'm deficient, and vitamin D as a staples. And most people are also deficient on those minerals. I tested my potassium, so I know that it's a little low, and also I use it a little bit for preventing stones. And perhaps you can say something about that, because my physician told me that yes, I have a family history, and potassium citrate seems to help also with that. So, but so far I've been taking this a long time, and I feel great. Yeah, omega-3s are, you know, I eat fish, but we just eat so the ratio omega-6 and omega-3 should be balanced in our body, and most of us eat too many omega-6s. I also, although I don't eat a processed diet, and omega-6s are found in vegetable oils, which are prevalent in processed food because they are cheap, and processed food wants to make money on. But I also eat nuts, a lot of nuts, and nuts are high in omega-6s. So I'm trying to balance my omega-6s and omega-3 fatty acids.
Yeah. Well, great. Well, thank you so much for taking the time to talk with us today. And I'm going to, any information you send me about new studies and desire for collaborators, we'll share with all our audience here and keep them in the loop. But once again, thanks so much for all the sharing all the exciting work you're doing. I don't know how you choose between such fascinating projects, but it was wonderful to have you on the show, Lucia. I really appreciate it.
Thank you very much. Thank you for having me. And just, I want to mention that I also have another passion, sharing my lectures on YouTube. Started recently. I just need a signal from people. If they like it, I will do more of that and sacrifice some weekends for that. But if not, I may just take my weekend off. So if you want to check out my YouTube channel and give me a like or subscription, that's appreciated. Thank you.
Yeah, we'll put the links to the YouTube channel in there, and I've checked it out. They're great presentations, very worth it. I highly recommend it. But thanks again, Lucia, and we look forward to chatting with you again on the show.
Thank you. Thank you for having me.
Note, this is not intended to be a substitute for professional medical advice, diagnosis, or treatment. Always seek the advice of your physician or other qualified health provider with any questions you may have. Never disregard professional medical advice or delay in seeking of it because of something you have seen here. If you find this to be of value of you, please hit that like button and subscribe to support the work we do on this channel. Also, we take your suggestions and advice very seriously. Please let us know what you'd like to see on this channel. Thanks for watching, and we'll hope to see you next time.